Hello, this is Dr. James Camp at Lee College in Baytown, Texas, and this is my presentation on the naming of, wait a minute, not cats, we're naming epithelial tissues. The joke here is there's a famous song in Andrew Lloyd Webber's musical Cats about the naming of cats, in which the cats tell you that Mr. Whiskers or Fluffy is not their true name. They have a true name that is only known by other cats that you, a mere human, will never know. But epithelia are not that pretentious. We're going to know the names of the epithelia by the time we're done with this short presentation. Okay, so epithelial tissues all have a 3 to 4 part name. Why 3 to 4? Well, there are always 3 parts, but sometimes there's a 4th part, a optional special feature name. So let's say we have a layer of cells. Remember, epithelial tissues are always tightly packed cells. And these cells happen to have little protrusions, cell hairs, called cilia on their apical surface. We would call that a ciliated epithelium. Fairly easy. Another one you'll see is perhaps we have a layered tissue here, and on top of that layer is a layer of dead cells filled with the protein keratin. This is what protects your epidermis from the world outside. So this tissue would get the special feature name keratinized. Okay, so the special feature name is fairly simple. It's not going to be found in all that many tissue names, but occasionally you'll find a ciliated tissue or a keratinized tissue. The first requirement is to know the number of layers. Okay, so again, let's say we have some cells and they're arranged in one layer going straight across. Here are their little nuclei, so we can know that they're cells. Or maybe they're arranged in a tubular shape, but there's only one layer of cells in this tube. The cell shape, or the layer type name for this, for a single layer, is simple. And that is for one layer of cells. The opposite of simple, you may have come across in your reading, is where we have multiple layers of cells stacked up, and in this case, we call the cell, the tissue, stratified. Strata is another, or stratum singular, is another of those Latin words, or it's a Latin word for layer. And so a stratified tissue has more than one layer. Okay. A simple way to know if you're dealing with a simple or a stratified tissue is if all the all the cells touch the basal lamina, then you have one layer of cells. It's simple. If there are cells that their basal layer is the apical layer of another cell, okay, then you have more than one layer, and that becomes a stratified tissue. Do 2 layers count as stratified? Yes. Do 10 layers count as stratified? Yes. It doesn't matter how many as long as it's more than one layer. All right, so let's move on. The second requirement that we know is a shape type. Okay, so cells can come, epithelial cells can come in basically one of 2 shapes. They can be, or one of 3 shapes. They can be wider, much wider than they are tall. That's essentially a flat shaped cell. Okay, a cell that looks like this. And the name for that is squamous. Okay, this is not what the word squamous actually means in Latin, but I think of the word squamous as being similar to the English word squashed. Okay, you've squashed the cell flat, so it's squamous. Alternatively, a cell can be much taller than it is wide. It can essentially be a very thin, tall cell. Okay. These resemble the columns on, if you've ever been out east of town and seen all the old pre-Civil War plantation mansions, they often have these columns running across the front porch. This resembles a column, so these are columnar, or you can also pronounce it columnar, but columnar epithelial cells. Well, what if you're neither really, really tall or really, really wide and flat? If you're sort of in the middle, you look kind of, the idea is that if you projected this cell into 3 dimensions, it would look sort of like a cube. So this cell is called cuboidal. Now I will warn you here that cuboidal cells very rarely look like a perfect square. Often they're part of a circular shape, and so they're maybe a little thinner at one edge and thicker at the other. And so cuboidal cells often have very odd shapes to them. The idea is, though, that they're not extremely wide and flat or extremely thin and tall. All right, so now we have the 2 required types, the layer type and the shape type. There's one last part. I promised you a 3-part name. There's one last required part, and that's the word epithelium. All you have to do for the third or fourth, if there's a special feature, all you have to do for that is write epithelium. So if I give you a lab test and there are 4 parts to the name of an epithelial tissue, you are guaranteed 25% credit if you just write epithelium in the last blank. With me? Good. Okay. All right, so now that we know the basic pieces of naming epithelia, let's look at the details of how to name a few sample tissues. So let's say we've got a little finger of intestine reaching out from your intestinal wall, and we notice that it's lined with cells that are very tall and thin. Okay. One important thing to do when you do this is notice where is, okay, this is the apical surface here, and this is the basal surface here. So when we're talking about cell shape, these are tall in this direction and not very wide in that direction. That makes these columnar cells. Okay, so we don't notice any special features about this tissue, so we're just going to go ahead and we're going to skip the special part of the name, and we're going to skip right on then to the layer type. There we see one layer here, so this is a simple. The shape is clearly columnar, and then we end by writing the word epithelium. Okay, see, that was fairly easy. Let's see if we can try another. Okay, here we have a circular tissue. Maybe it's a sweat gland. Okay, and we see that the cells here are not particularly shapely, but each one is neither particularly wide nor particularly tall. So we would guess that those would be they're about the same height as they are width. So these are cuboidal cells. Okay. Again, no special features, so we can skip the special section. Our layer type, how many layers do we have here? Well, first off, let's recognize again where our apical surface is. Okay. In any kind of a hollow organ, the lumen, the space on the inside, is going to be the apical surface, and then the part out here that's surrounded by tissue, that's going to be your basal surface. Okay, so we don't see any special features there. We do see that there are a couple of different layers between the apical and basal surfaces. So that means our layer type is going to be stratified. The shape we've already mentioned is cuboidal. And then we just need to tack in this word epithelium. Okay, so the full name of this tissue is a stratified cuboidal epithelium. One more easy example. Okay, we've got wisps of cells in your lung tissue, perhaps, that look like this. Each cell, you'll notice, is very wide and not very tall. So very flat, wide and flat. That would indicate squamous cell status. Okay, and we notice that there's really only one layer of these cells. So again, we can do away with the special, the layer type. There's only one layer, so it is simple. The shape type, these cells are wide and flat, so they are squamous. And then we end with the word epithelium again. So we have just named a simple squamous epithelium. All right, so you're getting the basic pattern. Let me throw a couple of curveballs your way. Here we're looking at an organ that has some odd looking cells to it. And if you look at the nuclei of these cells, you'll find that they assort themselves at various different layers. So this clearly looks like a tissue that has multiple layers. Furthermore, if we look at the apical surface of this tissue, we'll notice that there are little cell hairs sticking up. And in fact, we'll notice that there's a thin coating of mucus on those cell hairs. So we've got a lot going on here. First off, let's talk about the special feature. Okay, cell hairs are also known as cilia. So this tissue is ciliated. Now, what about the layer type? As I said, we've got multiple layers, levels of nuclei running across this. It makes it look like it is a multi-level tissue, but if you look carefully, every cell touches the basal layer, the basal lamina. So in truth, it's only one layer, but it looks like two layers, and we want to recognize that in some way. So the Latin word for something false, something that fakes you out, is pseudo. This is a pseudo stratified tissue. Okay. What about cell shape? We see some sort of same size, height and width cells down here at the bottom. We also see some tall and thin cells that reach all the way up to the top. How do we decide? The convention, for whatever reason, I wrote this in the wrong color. Silly me. The convention that tissue scientists, histologists use is that it's the cells that reach the top, the cells that reach the apical surface, that we use as a basis for this name. So we're going to go with the tall, thin cells and call this a columnar epithelium. So the full name for this tissue is a ciliated, pseudostratified columnar epithelium. Wow, that's a mouthful. Okay. But hopefully you see where we got that from. Now let's look at another kind of curveball example. We've got a tissue that has maybe multiple layers of cells. Down here, these cells look rather cuboidal. They're kind of jumbled in this intermediate zone. And then as they get higher up, they start spreading out into sort of brick-like cells. Okay, with more flattened nuclei. So we've got a lot going on here, but just to confuse things even more, we've got a layer of dead cells on top. You can tell they're dead because these don't have a particular, they don't have any organization, they don't have any nuclei in them to indicate their aliveness. Okay, so here on the apical surface, we've got a special feature. Anybody remember the name of that special feature? The dead cells are made of keratin or filled with keratin, so this is a keratinized epithelium. Layers, well, that one's pretty obvious. It should be pretty obvious that we have more than one layer here. So stratified. Now, what about the shape? Okay, we noticed again that we have cuboidal cells down here, but we have more brick-like, wider, flatter cells up here. What's the convention? We look at the apical surface. If we look at the cells up here at the apical surface, these cells are squamous. Okay, so the full name for this tissue is a keratinized, stratified squamous epithelium, also known, by the way, as your epidermis. Okay. One last example. Here we've got some cells that form a jumbled mess, really. Some of them are tall and thin, some of them are wide and flat. Some of them are about the same height and width. They have kind of a cobblestone look to them. But that's only one way that they look. Okay. When you distend this tissue, when you stretch it out to its maximum extent, now these cells look kind of more squamous, more organized, certainly a flatter layer of tissue. Okay. It's hard to identify in a tissue that has a variable number of layers and a variable cell shape, what to call that. How do we handle this? We don't say anything about the layers and we don't say anything about the shape. The full name for this tissue is simply the special feature transitional, because it transitions from one state back and forth to the other, and then the ending word epithelium. So the full name for this tissue is simply transitional epithelium. Okay. So that concludes our brief whirlwind tour of the naming of epithelia. You are now ready to go do your lab activity looking at actual real live epithelial slides and trying to determine the names for each of those. Good luck, and I'll see you in class.